The overall goal of this procedure is to automatically quantify morphological features of the Drosophila neuromuscular junction using morphometric software macros.
This method can help to identify regulators of synapse development, a key question in neurobiology. The main advantage of this technique is the automatic quantification of multiple NMJ features. This allows for objective analysis of NMJ morphology in high throughput.
We decided to develop this methodology to be able to investigate NMJ morphology in a large amount of disease models. We thought of ways how to prevent interpersonal differences and how to significantly speed up the quantification process.
Visual demonstration of this method is very helpful. Appropriate macro settings are crucial, and some steps might be difficult to new users, especially when not familiar with VG software.
For this protocol, generate image stacks of NMJs and save them as individual TIFF files, where channel 1 shows DLG1 staining, or a similar marker, and channel 2 shows BRP staining. To begin, create Z projections and hyperstacks of the NMJ image files. Open the plugin options and select Drosophila NMJ Morphometrics. Now, identify the unique file string that the microscope has assigned to the image series when storing them as TIFF. This will be at the end of the image name. Copy and paste the string given to the lowest plane and channel number into the unique file string setting window. Then, select the sub-macro convert.